So this chapter is on chapter 5. Don't be shy. You can't see your faces also. Chapter 5 is on the macromolecules. All right? And if you're paying attention, we covered 3 of them already. Does anyone remember what the 3 are? Any guesses?
Carbohydrates, lipids, and proteins.
Fantastic. So if you don't know these, make sure you have your notes or keep it in your head because I will be testing you on these next semester as well. Okay? So these are macromolecules, which mean large molecules. And all of these have what 3 elements for sure?
Carbon, hydrogen, oxygen.
Fantastic. Okay? And proteins, they're the only ones that
Marcus the science guy. Marcus the science guy. Marcus the science guy. Science rules. Marcus the science guy. Inertia is a property of matter. Marcus the science guy. Marcus, time under 10 seconds. Marcus the science guy.
Macromolecules. They're all around us. All around us. All around us. All around us. In your muscles, your skin, your DNA, your food, you name it. Macromolecules are essential to life. They all contain the main 3 elements: carbon, hydrogen, oxygen. Some of them also contain nitrogen and phosphorus. The 4 macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Let's start off with carbs. They're bread, pizza, chips, Oreos. Well, what really is a carb? The acronym for that would be CHO, or carbon, hydrogen, and oxygen. Bonds can be made and broken in 2 different ways. To be broken, that takes hydrolysis.
And to make a bond, you use dehydration reaction.
The most basic unit of a carbohydrate is a monosaccharide. Have 2, that's a disaccharide. Have 3, it's a polysaccharide. Here they are.
Alright guys, so if you did not understand what I was saying before, here's a nice visual representation of it. The first one is a monosaccharide. And then, see that little bind in between the second one? That's a glycosidic linkage. That's called a disaccharide. And then number 3, that's a polysaccharide. That's when there's 3 monosaccharides connected by glycosidic linkages. Now, do you remember how to break and make them? To make them, you use dehydration reaction. And to break them, you use hydrolysis. Alright guys, now it's time to move on to an experiment regarding carbohydrates. Here, you are going to watch an experiment regarding carbohydrates. First, label all of your test tubes by each number in numerical order. See? He labeled them 1 through 8. And then now here, for the test setup, in each different number, you're going to put 1 solution into it. For 1, water, 2, starch, 3, sucrose, 4, glucose, 5, apple, 6, coconut, 7, corn, and 8, soy. Here, you can see the guy putting half a drop into each test tube. See how he's putting coconut in the 6? That's exactly where he's supposed to. Now, add 10 drops of Benedict solution into each test tube. He's doing a great job right there. Exactly 10 drops. It's impressive. It's harder than it looks, guys. Trust me. I mean, look at that. 10 exactly. Wow. Alright, he's wrapping it up. And now, carefully place the entire test tube wrap into a very hot water bath. And you need to be careful because it's pretty hot. So use hot pads. 1 minute. 2 minutes. 3 minutes. After 3 minutes, pull the rack out of the water bath and let it cool for 1, 2, 3, 4, 5, 6, 7 minutes. And then, it's time to record your results. Clear blue means the absence of reducing sugars. The green-yellow means low levels of those reducing sugars. Here is a great way to visualize the results of this person's experiment. Go ahead and try it at home. It's pretty cool. Alright, that's it for carbs. Let's stop here for a second. I have a question for you. What do you call it when there's two monomers that are connected by glycosidic linkage? That's called a disaccharide. What's next? Lipids.
Lipids consist of the three elements: carbon, hydrogen, and oxygen. They have a high ratio of hydrogen to oxygen and consist of a bunch of bonds that help it store a lot of energy. Lipids consist of one glycerol and 3 fatty acids. There are 2 types of fatty acids: unsaturated fatty acids and saturated fatty acids. Here's my friend, the rock climber, Gabalicious Gabby, climbing a wall, and she will explain to you the difference between unsaturated fatty acids and saturated fatty acids.
Hey guys, this is Gabby. I'm going to show and tell you guys what unsaturated and saturated fats are. Okay, let's get cracking. Here we have unsaturated fat, or olive oil, and saturated fat, butter. So, what are the differences between these two types of fats? A saturated fat comes from animals, is solid at room temperature, and has a single bond. An unsaturated fat comes from plants, is liquid at room temperature, and has a double bond. This double bond allows the fat to be more fluid, and therefore is a big part of the semi-permeability of the cell membrane. Go. Okay. Now, let's get cooking with both of them. Right now, we're cooking an egg with an unsaturated fat, olive oil. Okay. Now, we're going to cook with some saturated fat. Okay, turn it off. Now that the pan is heated up, we're going to crack the egg. Oh my god, I messed that up.
I popped the yolk.
Sorry. Are you still filming?
Time lapse.
And where do unsaturated fats come from? Oh no, that's wrong. Saturated comes from salt and unsaturated comes from water. Just kidding. Saturated comes from animals and unsaturated comes from plants.
Now let's move into proteins.
I'm Oli, and I'm here to explain what a protein is. A protein consists of carbon, hydrogen, oxygen, and nitrogen. Well, the monomer of a protein is an amino acid. There are 20 different types, and each can either be classified as polar, nonpolar, or charged. Each are held together by a peptide bond to create one protein, and each has a Z group? No, a Y. Hey, Gabby!
What?
What's the group called?
What group?
The group of amino acids.
Oh, it's the R group.
So, how are proteins folded? Conformation. And what are the 4 different structures? First is the primary structure. This consists of just amino acids tied together by peptide bonds. And when the R groups interact, they create the secondary structure, which consists of alpha helixes and beta pleated sheets. And the tertiary structure, which is just a secondary structure folded up, it consists of 4 different kinds of bonds. There's the hydrogen bonds, the ionic bonds, the disulfide bridges, and the hydrophobic interactions. The quaternary structure is just 2 or more tertiary structures folded together.
Go.
Hey, I have a question for you. Wait, Marcus, get your finger up. They can't see me. Yeah, okay, that's much better. Anyways, as I was saying, what are the 3 groups we classify amino acids by? You got it. It's polar, nonpolar, and charged. Now let's move on to an experiment concerning proteins. Proteins are needed for muscle growth. They're found in our food and also baby food. Today, we will be testing how burette interacts with proteins. Usually, when it comes in contact with a protein, it will change from dark blue to dark purple. You'll be guessing which one of these baby foods has protein inside of it. One of them is made with strawberry and banana, and one of them is made with chicken and gravy. Which test tube do you think will have protein? Test tube A or test tube B? Now let's move on to the actual experiment. We're going to be putting 2 drops of burette into each test tube, and then we can see which one turns purple. Now that we have the results, were you right? Did you guess test tube A? This one turned purple, meaning that it contains proteins. This one did not turn purple, because the bananas and strawberries inside could not react with burette the same way proteins could.
And to make those proteins, you need nucleic acids. And the 2 nucleic acids are DNA,
double helix, double helix, double helix. Double helix, double helix, double helix. Double helix, double helix, double helix.
and RNA.
Single strand, 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 single strand.
Nucleic acids contain CHONP, or carbon, hydrogen, oxygen, nitrogen, phosphorus. The monomer of a nucleic acid is a nucleotide, and they contain a phosphate, sugar, and a base. And RNA is made up of a ribose sugar, which contain the... Wait. Hey, Gabby!
What?
What are the nitrogenous bases of RNA?
Oh, I know that. They're adenine, uracil, cytosine, and guanine.
All right, thanks, Gabby. And now, back to DNA. DNA contains the nitrogenous bases cytosine, adenine, guanine, and thymine. Those are held together by hydrogen bonds. DNA is also made up of a sugar, which is a deoxyribose sugar. And those nitrogenous bases are classified into two groups, apoptosis and photophosphorylation.
Marcus, you know that Mr. Nagiishi told us that it's purines and pyrimidines. Use the acronym PUGA to help you remember. Guanine and adenine are purines, whereas thionine and cytosine are pyrimidines.
Now it's time to move on to an experiment regarding one of the nucleic acids, DNA.
If only there was a good way to use genomics to find a good use for all these strawberries.
Are you thinking what I'm thinking? Oh yeah. DNA extraction! All you need is household items to set up. Otherwise, it'd take forever. Let's get started.
All you need is some dish soap, little cup, measuring cups, some glasses, plastic bags, some strawberries. They're looking pretty good. Some salts. Some chopsticks, or skewers, whatever you prefer. That's amazing. After this experiment, we'll be experts in strawberry and DNA.
Do you know how many copies of the genome each strawberry cell has?
I have no idea.
8! That's
8?
That's 4 times more per cell than humans. That's why purifying DNA is way easier than strawberries.
Whoa! Awesome.
It sure is awesome! No time to waste! Let's do some science!
Alright guys, now we need to make sure that we have everything that we need to extract some DNA from strawberries.
First, you need 2 strawberries.
Make sure to take the green part off. Next, 2 glasses.
Cheers. Some measuring cups.
Some measuring spoons.
A tablespoon of dish soap.
Some salt.
Half a cup of Tito's or rubbing alcohol.
Some chopsticks or skewers.
Half a cup of water.
A cloth, tissue, or coffee filter to make sure that you can drain. And a plastic bag. Maybe it can be reusable so you can help the environment. For the first step, you're going to put the 2 strawberries into your plastic bag. And then you're going to mash the strawberries. That way you can break down the DNA. Have a go, Justice. There you go. That's good.
And 1.
Next, we're gonna need 2 teaspoons of dish detergent. Alright, we do not even include all of this.
Yes, we do. We're gonna mute this.
Just put some music in the background. Some elevator music.
What are the... Okay, do I stop it?
Oh yeah, you're good.
Thumbs up, thumbs up, thumbs up.
Alright, the next step, you need to add your half cup of water. It's pretty full, so be careful. And then you need to swirl it. Uh-oh. That was an accident there. Make sure not to spill anything. We went back and fixed it. So instead, if you want, just mix it.
So this solution will be your extraction liquid. And what that will do, basically, these strawberry cells are like a water balloon. And this is the pin. So when you pop the water balloon, it will open up the strawberry cells. Next, we pour this solution in. Now we're going to want to seal it and gently massage the bag.
And then you start to gently massage the back.
Try not to form bubbles.
Just mute that part.
Come, take a look.
Wait, Justice. Our tissue just changed into a sifter. That's honestly a better option.
Yeah.
Alright, I would recommend using a sifter instead of a tissue. Alright, so here, now Justice, the next step, I want you to pour our solution.
I got
Through
you,
the
Marcus. We're gonna want to extract all this strawberry gunk out and just get the liquid. Here we go. Alright. Make sure not to spill it.
Alright, for your next step, you're going to pour your rubbing alcohol, or any hard liquor, into your solution.
Stop!
I got this for no worry, guys.
Next step, you're going to stir it a little bit. And then we're going to wait for the DNA to float to the top, and boom, that's your nucleic acid. Alright, we're going to let it rest for just a second. Oh, shoot. Justice, look at it forming. Wow. See that right there? It's pretty neat, huh? That's nucleic acid. And now, I'm gonna go on to my friend Justice, who knows a lot more about DNA than I do. And he's gonna talk about something called whatever, let's go through it.
I got you, Marcus. DNA undergoes a very special process called semi-conservative replication. This was discovered by Messelson and Stahl.
Semi-conservative replication is a complex process occurring during S phase of interphase. All cells need to undergo this process to grow and reproduce. It involves the separation of the DNA strands and the synthesis of 2 new complementary strands. So, let's go into how this process works. First, DNA helicase will unwind a DNA strand. Then, SSBP will bind to each DNA strand to keep them apart from one another. RNA primer will now bind to both strands to allow DNA to begin copying itself with DNA polymerase. It's important to note that during this step, the 2 strands undergo different steps. The top strand, which ends on a 3' end, is copied on one piece. This is because DNA copies from 5' to 3'. This strand is called the leading strand. On the bottom strand, however, DNA is unable to copy into one big piece because it ends with a 5' end. Thus, RNA primer and DNA polymerase work together to create small strands as the helicase unwinds. These strands are called the Okazaki fragments. DNA ligase then comes in and joins these fragments together. These steps are all the steps for DNA replication. As a result of semi-conservative replication, each new DNA molecule consists of 1 original parental strand and 1 newly synthesized daughter strand. This mechanism ensures the preservation of this genetic information during cell division and inheritance.
Hey guys, I have a question for you. What keeps the DNA strands apart at the beginning of semi-conservative replication? SSBP, single-stranded binding proteins.
Alright guys, that concludes the awesome video on macromolecules.
Hope you enjoyed. Thank you for watching.
Yeah, alright, and
Yeah. Cue the outro.
before we go guys, do you know enough about macromolecules? If not,
Ask me these questions.
yeah, but again, cue the outro. And thank you for watching our presentation.